Hello friends, welcome to my Sakshi Coaching channel. Now in this video, I am going to read the chapter 14 in the biology class 11th, respiration in plants. So let's start. All of us breathe to live, but why is breathing so essential to life? What happens when we breathe? Also, do all living organisms including plants and microbes breathe? If so, how? All living organisms need energy for carrying out daily life activities, be it absorption, transport, movement, reproduction, or even breathing. Where does all this energy come from? We know that we eat food for energy, but how is this energy taken from food? How is this energy utilized? Do all foods give the same amount of energy? Do plants eat? Where do plants get their energy from? And microorganisms, for their energy requirement, do they eat food? You may wonder at several questions raised above. They may seem to be very disconnected. But in reality, the process of breathing is very much connected to the process of release of energy from food. Let us try and understand how this happens. All energy required for life processes is obtained by the oxidation of some macromolecules that we call food. Only green plants and cyanobacteria can prepare their own food by the process of photosynthesis. They trap light energy and convert it into chemical energy that is stored in the bonds of the carbohydrates like glucose, sucrose, and starch. We must remember that in green plants too, not all cells, tissues, and organs photosynthesize. Only cells containing chloroplast that are most often located in superficial layers carry out photosynthesis. Hence, even in green plants, all other organs, tissues, and cells that are non-green need food for oxidation. Hence, food has to be translocated to all the non-green parts. Animals are heterotrophic, that is, they obtain food from plants directly, herbivores, or indirectly, carnivores. Saprophytes like fungi are dependent on dead and decaying matter. What is important to recognize is that ultimately all the food that is required for the life processes comes from photosynthesis. This chapter deals with the cellular respiration, or the mechanism of the breakdown of the food materials within the cell to release energy and the trapping of this energy for synthesis of ATP. Photosynthesis, of course, takes place within the chloroplast in the eukaryotes, whereas the breakdown of the complex molecules to yield energy takes place in the cytoplasm and in the mitochondria, also only in eukaryotes. The breaking of the CC bonds of the complex compounds through oxidation within the cells, leading to a release of considerable amount of energy, is called respiration. The compounds that are oxidized during this process are known as respiratory substrates. Usually, carbohydrates are oxidized to release energy, but proteins, fats, and even organic acids can be used as respiratory substances in some plants under certain conditions. During oxidation within a cell, all the energy contained in respiratory substrates is not released free into the cell or in a single step. It is released in a series of the slow stepwise reactions controlled by the enzymes, and it is trapped as chemical energy in the form of ATP. Hence, it is important to understand that the energy released by the oxidation in respiration is not, or rather cannot be used directly, but is used to synthesize ATP, which is broken down whenever and wherever energy needs to be utilized. Hence, ATP acts as the energy currency of the cell. This energy trapped in ATP is utilized in various energy requiring processes of the organism, and carbon skeleton produced during the respiration is used as a precursor for biosynthesis of other molecules in the cell. 14.1 Do plants breathe? Well, the answer to this question is not quite so direct. Yes, plants require oxygen for respiration to occur, and they also give out CO2. Hence, plants have systems in place that ensure the availability of O2. Plants, unlike animals, have no specialized organs for gaseous exchange, but they have stomata and lenticels for this purpose. There are several reasons why plants can get along without respiratory organs. First, each plant part takes care of its own gas exchange needs. There is very little transport of gases from one plant part to the another. Second, plants do not present great demands for gas exchange. Roots, stems and leaves respire at rates far lower than the animals do. Only during photosynthesis are large volumes of the gases exchanged, and each leaf is well adapted to take care of its own needs during these periods. When cells photosynthesize, availability of O2 is not a problem in their cells since O2 is released within the cell. Third, the distance that gases must diffuse even in large, bulky plants is not great. Each living cell in a plant is located quite close to the surface of the plant. This is true for leaves. You may ask, but what about thick, woody stems and roots? In stems, the living cells are organized in thin layers inside and beneath the bark. They also have openings called lenticels. The cells in the interior are dead and provide only mechanical support. Thus, most cells of a plant have at least a part of their surface in contact with air. This is also facilitated by the loose packing of parenchyma cells in leaves, stems and roots, which provide an interconnected network of air spaces. The complete combustion of glucose, which produces CO2 and H2O as end products, yields energy, most of which is given out as heat. C6H12O6 plus CO2 gives out 6CO2 plus 6H2O plus some energy. If this energy is to be useful to the cell, it should be able to utilize it to synthesize other molecules that the cell requires. The strategy that this plant cell uses is to catabolize the glucose molecule in such a way that not all the liberated energy goes out as heat. The key is to oxidize glucose not in one step but in several small steps, enabling some steps to be just large enough such that the energy released can be coupled to the ATP synthesis. How this is done is essentially the story of respiration. During the process of respiration, oxygen is utilized and carbon dioxide, water and energy are released as products. The combustion reaction requires oxygen, but some cells live where oxygen may or may not be available. Can you think of such situations and organisms where oxygen is not available? There are sufficient reasons to believe that the first cells on this planet lived in atmosphere that lacked oxygen. Even among the present day living organisms, we know of several that are adapted to anaerobic conditions. Some of these organisms are facultative anaerobes, while in others, the requirement for anaerobic condition is obligate. In any case, all living organisms retain the enzymatic machinery to partially oxidize glucose without the help of oxygen. This breakdown of glucose to pyruvic acid is called glycolysis. 14.2 Glycolysis. The term glycolysis has originated from the Greek words glykos for sugar and lysis for splitting. The scheme of glycolysis was given by Gustav Emden, Otto Merov and J. Parnas and is often referred to as EMP pathway. In anaerobic organisms, it is the only process in respiration. Glycolysis occurs in the cytoplasm of the cell and is present in all living organisms. In this process, glucose undergoes partial oxidation to form two molecules of pyruvic acid. In plants, this glucose is derived from the sucrose, which is the end product of the photosynthesis, or from storage carbohydrates. Sucrose is converted into glucose and fructose by the enzyme invertase, and these two monosaccharides readily enter the glycolytic pathway. Glucose and fructose are phosphorylated to give rise to the glucose 6-phosphate by the activity of the enzyme hexokinase. This phosphorylated form of the glucose then isomerizes to produce fructose 6-phosphate. Subsequent steps of the metabolism of glucose and fructose are same. The various steps of the glycolysis are depicted in the figure 14.1. In glycolysis, a chain of 10 reactions under the control of different enzymes takes place to produce pyruvate from glucose. While studying the steps of the glycolysis, please note the steps at which utilization ATP energy or synthesis of ATP, or in this case of NADH plus H plus, take place. ATP is utilized at two steps. First in the conversion of glucose into glucose 6-phosphate, and second in the conversion of the fructose 6-phosphate to fructose 1,6-diphosphate. The fructose 1,6-diphosphate is split into dihydroxyacetone phosphate and 3-phosphoglyceraldehyde. We find that there is one step where NADH plus H plus is formed from NAD plus. This is when 3-phosphoglyceraldehyde PGAL is converted to the 1,3-bisphosphoglycerate DPGA. Two redox equivalents are removed in the form of the two hydrogen atoms from PGAL and transferred to a molecule of NAD+. PGAL is oxidized and with inorganic phosphate to get converted into DPGA. The conversion of the DPGA to 3-phosphoglyceric acid PGA is also an energy yielding process. This energy is trapped by the formation of ATP. Another ATP is synthesized during the conversion of PEP to pyruvic acid. Can you then calculate how many ATP molecules are directly synthesized in the pathway from one glucose molecule? Pyruvic acid is then the key product of glycolysis. What is the metabolic fate of pyruvate? This depends on the cellular need. There are three major ways in which different cells handle pyruvic acid produced by glycolysis. These are lactic acid fermentation, alcoholic fermentation, and aerobic respiration. Fermentation takes place under the anaerobic conditions in many prokaryotes and unicellular eukaryotes for the complete oxidation of the glucose to CO2 and H2O. However, organisms adopt a Krebs cycle, which is also called as aerobic respiration. This requires oxygen supply. 14.3 Fermentation. In fermentation, say by yeast, the incomplete oxidation of glucose is achieved under the anaerobic conditions by the sets of reactions where pyruvic acid is converted to CO2 and ethanol. The enzymes pyruvic acid decarboxylase and alcohol dehydrogenase catalyze these reactions. Other organisms like some bacteria produce lactic acid from pyruvic acid. The steps involved are shown in the figure 14.2. In animal cells, also like muscles during exercise, when oxygen is inadequate for cellular respiration, pyruvic acid is reduced to lactic acid by lactate dehydrogenase. The reducing agent is NADH plus H plus, which is reoxidized to NAD plus in both the processes. If both lactic acid and alcohol fermentation, not much energy is released. Less than 7% of the energy in glucose is released and not all of it is trapped as high energy bonds of ATP. Also, the processes are hazardous, either acid or alcohol is produced. What is the net ATPs that is synthesized? Calculate how many ATP are synthesized and deduct the number of the ATP utilized during glycolysis when one molecule of the glucose is fermented to alcohol or lactic acid. Yeast poison themselves to death when the concentration of the alcohol reaches about 13%. What then would be the maximum concentration of the alcohol in beverages that are naturally fermented? How do you think alcoholic beverages of alcohol content greater than this concentration are obtained? What then is the process by which organism can carry out complete oxidation of glucose and extract the energy stored to synthesize a larger number of the ATP molecules needed for the cellular metabolism? In eukaryotes, these steps take place within the mitochondria and this requires O2. Aerobic respiration is the process that leads to complete oxidation of organic substances in the presence of oxygen and releases CO2, water and a large amount of energy present in the substrate. This type of respiration is most common in higher organisms. We will look at these processes in the next section. 14.4 Aerobic respiration. For aerobic respiration to take place within the mitochondria, the final product of the glycolysis, pyruvate, is transported from cytoplasm into the mitochondria. The crucial events in aerobic respiration are the complete oxidation of the pyruvate by the stepwise removal of all the hydrogen atoms, leaving 3 molecules of CO2. Second, the passing on of the electrons removed as a part of the hydrogen atoms to molecular oxygen with simultaneous synthesis of ATP. What is interesting to note that is the first process takes place in the matrix of the mitochondria, while the second process is located on the inner membrane of the mitochondria. Pyruvate, which is formed by the glycolytic catabolism of carbohydrates in the cytosol, after it enters mitochondrial matrix, undergoes oxidative decarboxylation by a complex set of reactions catalyzed by the pyruvic dehydrogenase. The reactions catalyzed by the pyruvic dehydrogenase require the participation of several coenzymes including NAD plus and coenzyme A. Pyruvic acid plus coenzyme A plus NAD plus in the presence of the magnesium plus 2 pyruvate dehydrogenase gives out acetyl coenzyme A plus CO2 plus NADH plus H plus. During this process, the two molecules of the NADH are produced from the metabolism of the two molecules of the pyruvic acid produced from one glucose molecule during glycolysis. The acetyl coenzyme A then enters a cyclic pathway tricarboxylic acid cycle, more commonly called as Krebs cycle, after the scientist Hans Krebs who first elucidated it. 14.4.1 Tricarboxylic acid cycle. The TCA cycle starts with the condensation of the acetyl group with oxaloacetic acid and water to yield the citric acid. The reaction is catalyzed by the enzyme citrate synthase and molecule of coenzyme A is released. Citrate is then isomerized to isocitrate. It is followed by the two successive steps of decarboxylation leading to the formation of alpha ketoglutaric acid and then succinyl coenzyme A. In the remaining steps of the citric acid cycle, succinyl coenzyme A is oxidized to the oxaloacetic acid, allowing the cycle to continue. During the conversion of the succinyl coenzyme A to succinic acid, a molecule of GTP is synthesized. This is a substrate level phosphorylation. In a coupled reaction, GTP is converted to GDP with the simultaneous synthesis of ATP from ADP. Also, there are 3 points in the cycle where NAD plus is reduced to the NADH plus H plus and 1 point where FAD plus is reduced to the FADH2. The continued oxidation of the acetic acid via the TCA cycle requires the continued replacement of oxaloacetic acid, the first member of the cycle. In addition, it also requires regeneration of NAD plus and FAD plus from NADH and FADH2 respectively. The summary equation for this phase of respiration may be written as follows: pyruvic acid plus 4 NAD plus FAD plus 2H2O plus ADP plus inorganic phosphate in the mitochondrial matrix gives out 3 CO2 plus 4 NADH plus 4 H plus FADH2 plus ATP. We have till now seen that glucose has been broken down to release CO2 and 8 molecules of NADH plus H plus. 2 of FADH2 have been synthesized besides just 2 molecules of ATP. You may be wondering why we have been discussing respiration at all. Neither oxygen has come into the picture nor the promised large number of ATP has yet been synthesized. Also, what is the role of the NADH plus H plus and FADH2 that is synthesized? Let us now understand the role of oxygen in the respiration and how ATP is synthesized. 14.4.2 Electron Transport System ETS and Oxidative Phosphorylation. The following steps in the respiratory process are to release and utilize the energy stored in the NADH plus H plus and FADH2. This is accomplished when they are oxidized through the electron transport system and the electrons are passed on to oxygen, resulting in the formation of H2O. The metabolic pathway through which the electron passes from one carrier to the another is called the electron transport system ETS, and it is present in the inner mitochondrial membrane. Electrons from NADH produced in the mitochondrial matrix during the citric acid cycle are oxidized by an NADH dehydrogenase complex 1 and electrons are then transferred to ubiquinone located within the inner membrane. Ubiquinone also receives reducing equivalents via FADH2 complex 2 that is generated during the oxidation of succinate in the citric acid cycle. The reduced ubiquinol is then oxidized with the transfer of the electrons to cytochrome C via cytochrome BC1 complex 3. Cytochrome C is a small protein attached to the outer surface of the inner membrane and act as a mobile carrier for transfer of electrons between complex 3 and 4. Complex 4 refers to the cytochrome C oxidase complex containing cytochromes A and A3 and the two copper centers. When the electrons pass from one carrier to the another via complex 1 to 4 in the electron transport chain, they are coupled to ATP synthase complex 5 for the production of ATP and ADP and inorganic phosphate. The number of the ATP molecules synthesized depends on the nature of the electron donor. Oxidation of one molecule of NADH gives rise to the 3 molecules of ATP, while that of one molecule of FADH2 produces 2 molecules of ATP. Although the aerobic process of respiration takes place only in the presence of the oxygen, the role of oxygen is limited to the terminal stage of the process. Yet the presence of the oxygen is vital since it drives the whole process by removing hydrogen from the system. Oxygen acts as the final hydrogen acceptor. Unlike photophosphorylation, where it is the light energy that is utilized for the production of proton gradient required for the phosphorylation, in respiration it is the energy of the oxidation reduction utilized for the same process. It is for this reason that the process is called oxidative phosphorylation. You have already studied about the mechanism of the membrane linked ATP synthesis as explained by the chemiosmotic hypothesis in the earlier chapter. As mentioned earlier, the energy released during the electron transport system is utilized in synthesizing ATP with the help of ATP synthase complex 5. This complex consists of two major components F1 and F0. Figure 14.5. The F1 headpiece is a peripheral membrane protein complex and contains the site for synthesis of ATP from ADP and inorganic phosphate. F0 is an integral membrane protein complex that forms the channel through which protons cross the inner membrane. The passage of protons through the channel is coupled to the catalytic site of the F1 component for the production of ATP. For each ATP produced, 2H plus passes through the F0 from the intermembrane space to the matrix down the electrochemical proton gradient. 14.5 The respiratory balance sheet. It is possible to make calculations of the net gain of ATP for every glucose molecule oxidized, but in reality this can remain only a theoretical exercise. These calculations can be made only on certain assumptions that there is a sequential orderly pathway functioning with the one substrate forming the next and with glycolysis, TCA cycle and ETS pathway following one after the another. The NADH synthesized in glycolysis is transferred into the mitochondria and undergoes oxidative phosphorylation. None of the intermediates in the pathway are utilized to synthesize any other compound. Only glucose is being respired, no other alternative substrates are entering in the pathway at any of the intermediary stages. But these kind of assumptions are not really valid in a living system. All pathways work simultaneously and do not take place one after the another. Substrates enter the pathways and are withdrawn from it, and when necessary, ATP is utilized as and when needed. Enzymatic rates are controlled by multiple means. Yet it is useful to do this exercise to appreciate the beauty and efficiency of the living system in the extraction and storing energy. Hence, there can be a net gain of 36 ATP molecules during aerobic respiration of one molecule of glucose. Now let us compare fermentation and aerobic respiration. Fermentation accounts for only a partial breakdown of glucose, whereas in aerobic respiration it is completely degraded to CO2 and H2O. In fermentation there is a net gain of only 2 molecules of ATP for each molecule of glucose degraded to pyruvic acid, whereas many more molecules of the ATP are generated under aerobic conditions. NADH is oxidized to the NAD plus rather slowly in fermentation. However, the reaction is very vigorous in case of aerobic respiration. 14.6 Amphibolic pathway. Glucose is favored substrate for respiration. All carbohydrates are usually first converted into glucose before they are used for respiration. Other substrates can also be respired as has been mentioned earlier, but then they do not enter the respiratory pathway at the first step. See figure 14.6 to see the points of the entry of different substrates in the respiratory pathway. Fats would need to be broken down into glycerol and fatty acids first. If fatty acids were to be respired, they would first be degraded to acetyl coenzyme A and enter the pathway. Glycerol would enter the pathway after being converted to PGAL. The proteins would be degraded by the proteases and the individual amino acids after deamination, depending on their structure, would enter the pathway at some stage within the Krebs cycle or even as pyruvate or acetyl coenzyme A. Since respiration involves breakdown of the substrates, the respiratory process has traditionally been considered a catabolic process and the respiratory pathway as a catabolic pathway. But in this understanding correct? We have discussed above at which points in the respiratory pathway different substrates would enter if they were to be respired and used to derive energy. What is important to recognize is that it is these very compounds that would be withdrawn from the respiratory pathway for the synthesis of the said substrates. Hence, fatty acids would be broken down to acetyl coenzyme A before entering the respiratory pathway when it is used as a substrate. But when the organism needs to synthesize fatty acids, acetyl coenzyme A could be withdrawn from the respiratory pathway for it. Hence, the respiratory pathway comes into the picture both during breakdown and the synthesis of the fatty acids. Similarly, during breakdown and synthesis of the protein too, respiratory intermediates form the link. Breaking down processes within the living organism is catabolism and the synthesis is anabolism. Because the respiratory pathway is involved in both anabolism and catabolism, it would hence be better to consider the respiratory pathway. Figure 14.6 Interrelationship among the metabolic pathways showing respiration mediated breakdown of different organic molecules to CO2 and water. As an amphibolic pathway rather than a catabolic one. 14.7 Respiratory quotient. Let us now look at another aspect of respiration. As you know, during the aerobic respiration, O2 is consumed and CO2 is released. The ratio of the volume of CO2 evolved to the volume of the O2 consumed in the respiration is called respiratory quotient or respiratory ratio. RQ equals to volume of CO2 evolved upon volume of O2 consumed. The respiratory quotient depends upon the type of the respiratory substrate used during respiration. When carbohydrates are used as a substrate and are completely oxidized, the RQ will be 1 because equal amounts of CO2 and O2 are evolved and consumed respectively, as shown in the equation below. C6H12O6 plus 6O2 gives out 6 CO2 plus 6 H2O plus energy. RQ equals to 6 CO2 upon 6 O2 equals to 1. When fats are used in the respiration, the RQ is less than 1. Calculations for a fatty acid, try palmitin, if used as a substrate is shown. To C51H98O6 plus 145 O2 gives out 102 CO2 plus 98 H2O plus energy. RQ equals to 102 CO2 and 145 O2 equals to 0.7. When proteins are respiratory substrates, the ratio would be about 0.9. What is important to recognize is that in living organisms, respiratory substances are often more than one. Pure proteins or fats are never used as a respiratory substrates. Summary. Plants, unlike animals, have no special systems for breathing or gaseous exchange. Stomata and lenticels allow gaseous exchange by diffusion. Almost all living cells in a plant have their surfaces exposed to air. The breaking of the CC bond of complex organic molecules by oxidation cells, leading to the release of a lot of energy, is called cellular respiration. Glucose is favored substrate for respiration. Fats and proteins can also be broken down to yield energy. The initial stage of the cellular respiration takes place in the cytoplasm. Each glucose molecule is broken through a series of the enzyme catalyzed reactions into two molecules of the pyruvic acid. This process is called glycolysis. The fate of the pyruvate depends upon the availability of oxygen and the organism. Under anaerobic conditions, either lactic acid fermentation or alcohol fermentation occurs. Fermentation takes place under aerobic conditions in many prokaryotes, unicellular eukaryotes, and in germinating seeds. In eukaryotic organisms, aerobic respiration occurs in the presence of oxygen. Pyruvic acid is transported into the mitochondria, where it is converted into acetyl coenzyme A with the release of CO2. Acetyl coenzyme A then enters the tricarboxylic acid pathway or Krebs cycle, operating in the matrix of the mitochondria. NADH plus H plus and FADH2 are generated in the Krebs cycle. The energy in these molecules as well as that in the NADH plus H plus synthesized during the glycolysis are used as to synthesize ATP. This is accomplished through a system of electron carriers called electron transport system ETS located on the inner membrane of the mitochondria. The electrons, as they move through the system, release enough energy that are trapped to synthesize ATP. This is called oxidative phosphorylation. In this process, O2 is ultimate acceptor of electrons and it gets reduced to water. The respiratory pathway is an amphibolic pathway as it involves both anabolism and catabolism. The respiratory quotient depends upon the type of the respiratory substance used during respiration. Chapter completed. If you like this video, please like, share and subscribe to my channel to get all updates and more videos. Thank you.